Thank you very much for attending. I would like to ask our Dean, Mike Trick, to introduce the guest today for the talk.
Well, thank you all. And welcome to Carnegie Mellon University in Qatar. My name's Michael Trick. I'm the dean here. And so at this campus, we are privileged to have many scholars and experts visit each year to share their expertise. Many are from our campus in Pittsburgh. We created the Distinguished Lecture Series to showcase the depth and breadth of scholarship of these visitors. Distinguished Lectures in Computer Science are named in honor of Nico Habermann. Dr. Habermann was a pioneer in computer science and the founding dean of CMU's School of Computer Science, which of course is the world leader in AI research. It's my honor to introduce our speaker today to talk about the evolving role of computation in biomedical discovery. Dr. Russell Schwartz is a professor of computational biology and biological sciences at CMU. He is also head of the named Ray and Stephanie Lane Computational Biology Department within the School of Computer Science. Dr. Schwartz's research has covered many applications of modeling, simulation, algorithms, and machine learning in biology, including work in computational genomics, phylogenetics, population genetics, and biophysics. He has also worked to improve the practice of bioinformatics in education, collaborating with the global education community to find best practices for computational training in the life sciences. Please join me in welcoming Dr. Russell Schwartz to the Distinguished Lecture Series.
Thank you for having me here, and thank you for the invitation. I'm really enjoying my visit to the CMU Qatar campus, and I wanted to tell you a bit about some of my perspective on how the role of computation in biomedical discovery has been evolving over the course of my career and where I think it might be going in the future. So a talk like this necessarily has to be based on my own perspective, so I wanted to give a little bit of background on where I'm drawing these conclusions from. So what have I done as a scientist, and how does that inform my thinking, and some of the things I hope to share with you about where I think computation in biomedical science has been going and where it's likely to be going in the future. And I'll be sharing my perspective in part as a scientist who's been working at the intersection of computation and biology for a number of years, as an administrator who's been working in various capacities on managing computational biology science beyond my own research agenda, and as an educator who's been working to teach people in this space, both computational biologists and experimental scientists, and working with the global education community there. And I hope this will give me some perspective that I can share with you about why I think it's so important for anyone working in either of these fields, life sciences or computational sciences, to know about work at the intersection between them. So I have had the privilege of working in many different areas of biology over the course of my career. I started in problems more or less of a biophysical modeling character, moved in postdoctoral work to other kinds of biophysical sciences, went after my postdoctoral work to industry for a few years, where I got into problems of genome assembly and sequence analysis, brought both of those to Carnegie Mellon a few years after that, along with various other problem areas that I got into from there, branching off into problems in population genetics, phylogenetics, as well as bringing the biophysical work into new areas in cell biology, biomechanics, model fitting, so learning models of biochemical data, and so forth. And I will say for any students there, it is not something that would generally be recommended that you try to work on so many different things. It's probably not a good thing for your career to do this, but I'm very glad I did, because it's given me a chance to see many different fields, and not just different fields of biology, but different fields of computing. because in getting into all of these areas, I've learned about many different computational paradigms that end up applying to these. So I've done work that involves computing from computational chemistry, computational physics, statistical computing, numerical computing, discrete algorithms, operations research, and AI and machine learning, all of which end up feeding into different aspects of these problems. And I think that's given me a perspective from which I've been able to observe how different aspects of this field have evolved that I hope will allow me to give some guidance of where I think they're likely to be going later. And one of the things I have seen from many of these different areas is just over the course of my career in computational biology, which is about 30 years at this point, how much has changed in terms of the importance of computation in biology and how much that's appreciated from both the biological and life sciences perspectives. To give one anecdote of this, some of my work was in the area of protein folding, so looking at problems of how proteins find their three-dimensional structure. And this is an area in which, at the time I got into it, I would say protein folding was its own niche area of computational people and theoreticians that did not get a lot of respect from most experimental biologists. It was largely regarded as something that had no particular relevance to how actual protein folding works, and that was probably a fair assessment 30 years ago, because the models were too crude to really be able to describe how single proteins folded. There are certainly exceptions. My postdoctoral advisor, Jonathan King, was a brilliant experimental protein biochemist who appreciated that computation was going to be important to the field, but that was probably not a common opinion among his colleagues at the time. If we look at what's happened since then, protein folding went from something that basically had nothing to say about how actual proteins folded, aside from very broad general principles, to something that occasionally worked for describing real proteins, then often worked, and today is largely trusted as a way of studying how actual proteins fold. Many of you have probably heard of AlphaFold, which has really revolutionized the field, but it's important to know that AlphaFold is the end point, or really not even the end point, one more point in a long field of discoveries that have built upon one another, leading most recently a few weeks ago to the recent Nobel Prize for AlphaFold and some earlier work in this area. So what all of that is getting at is, I think, really a tremendous change in the importance of computation as it's seen within one little niche of this field. And while that's one particularly dramatic example, I don't think it's appreciably different from what I've seen in any of these other fields I've worked in. Every area of biology today understands that they need computation. And this was not true when I was starting as a computational biologist 30 years ago, and I think it's only going to become more true with time. Today, I work largely in cancer biology, and I would argue that this is a great field to be in if you're a computational biologist, in part because it's a very important area. No one is immune to cancer. Every population on Earth is affected by cancer. Everyone probably knows someone who has been affected by cancer. And so it's an area where computation can have a great impact, but it's also an area that in many ways is a microcosm of the whole field. So just as I mentioned, working in many different areas of biology, pretty much any area of biology you can think of has some relevance to cancer biology. As someone working in cancer biology, I do work that touches on cell biology, biophysics, genetics, genomics, medical biology, as well as many other areas such as biomedical engineering, bioimaging, and numerous areas of computing. So just within cancer biology, I once again do work that involves discrete algorithms, AI and machine learning, operations research, statistical computing, pretty much any paradigm of computing, any paradigm of biology you can think of comes up in cancer biology. But it's also a really interesting field for computational biology because it tends to be at the cutting edge of pretty much everything that's happening at the intersection of experimental and computational biology. Biology has been transformed by new technologies again and again, introducing new ways of studying biology to the field, new kinds of genomic methods like single-cell genomics, now spatial genomics. Almost all of these initially find some of their greatest application in cancer biology. And it's been at that intersection of these fields, where biomedical engineering and biotechnology are influencing biology, that computational biology often has its greatest impact. And this is another place where I think it's been very exciting to sit in computational biology and to see where the field is going, because often what's happening in cancer biology is a forerunner for what's going to be happening in any other field of biology. And so I want to tell you a little bit about what I'm doing and what I think the field is doing in the area of computational cancer biology, because I think it has important lessons for where any kind of biology you can think of is going to be going in the future. So one of the things that I am working on, and many people in the field are working on, is keeping up with these constant changes in technology. Cancer biology has been very heavily driven by ways of studying genetic variations within a tissue. So when we study cancer biology, to a large degree, we're studying a process of evolution. We're studying how genetic variations accumulate in developing tumors and how that accumulation leads to the progression of cancers. And so this is a field that has been driven by continuing advances in our ability to study genetic variation, from pre-genomic technologies like FISH, which is fluorescence in situ hybridization, essentially an image-based technology for looking at very small numbers of variations, to bulk DNA sequencing, to single-cell sequencing, and today to more advanced technologies such as spatial genomics. The field is continually advancing, and those of us in computation have to continually advance concurrent with this because the technology is ever changing. More and more, what is going on in cancer biology is being driven by a cycle of the science, the scientific discovery following advances in the technology. And often that is advances in biotechnology for studying biological systems, and more often it's advances in the computational technology, and these feed into one another. So as new technologies come along for profiling DNA sequencing, new computational technologies need to keep up with this. And those new computational technologies are often suggesting new directions of research that then lead to new advances in biotechnology. And more often what's happening in scientific discovery is scientific questions following the technology rather than the reverse. Not only that, but from a computational perspective, it's becoming more and more complicated because every time a new technology comes along, it turns out to have some interesting new things it can show you, some advantages, but it never completely displaces the technologies that came before. When bulk sequencing came along, it was really exciting, allowing us to study entire genomes at once, but it didn't completely replace the earlier pre-genomic technologies that had some advantages in studying what was happening at the single-cell level. Single-cell sequencing had some substantial advantages in cancer work over bulk sequencing, allowing us to look at the level of resolution of single cells, but also disadvantages. And as computational work came along to work with each of these technologies, we could not just move on to a new technology. It's not just about keeping up with each new technology. We also had to bring in all of the old technologies. So the more the technology advances, the more complicated the portrait becomes, because each of these old technologies is still telling us something important about how a cancer develops. This is just an example showing how we're using 3 different generations of technology for studying genetic variations in DNA and using a kind of constrained optimization framework to integrate all of these into a more complete portrait and essentially show that having all of these allows you to do a better job of figuring out what's happening in tumor development than if we looked at any 1 or 2 of these alone. And that is a trend that is continuing as the technologies advance. My lab and many others in the field, for example, are quite excited today about blood-based sequencing, another one of these technologies that is continually advancing, becoming important, well, it's already important for cancer studies and beginning to become important as a window into other kinds of problems in biology. Blood-based sequencing is a way of studying tumor DNA from DNA that is secreted from the tumor into the bloodstream, and it gives us yet another window into what's happening in tumor development. By combining tissue-based sequencing, looking at the tumor itself, with the blood-based sequencing, each of these has some advantages that allow us to get, once again, a more complete picture of tumor development. The tissue allows us to do a better job of constructing what's happening in evolution of the tumor, discovering populations of clonal cells that develop as the tumor is evolving. The blood-based sequencing gives us the ability to look at finer temporal resolution, to be able to look at different time points in evolution of a tumor and understand what's happening within them. But it's really the combination of the two that gives you power to be able to tell a story about how a tumor is developing in much finer detail. In this case, this is an example of a picture we can put together from a data set taken from a single cancer patient, reconstructing an evolutionary tree, but also telling a story, in this case an unfortunately sad story about this patient. A patient who was treated for cancer began to show evidence of recurrence of their tumor through rise of one of the clones, one of the cancerous cell populations. We could see that taking off as other clones expanded, and then lead to a change in treatment strategy to treat those clones that had expanded here. But unfortunately, as those clones responded to a new treatment, another clone shot up resistant to that treatment, and this would have been the end point for the patient. We can't be certain since this wasn't our patient, but most likely this led to the mortality of the patient. This kind of story is about more than just reconstructing what happened in the past. It's about being able to understand why these things happened and being able to start to develop methods that can do a better job of changing the treatment paradigm as we progress. Being able to say earlier that that patient was at risk of recurrence, maybe beginning to recur, and perhaps being able to say earlier how they might have been treated differently to head off that relapse with a resistant cell population. And this is just one aspect of something that is going on at many different levels in cancer research, and that I think is a forerunner of what's going to be happening more and more in any kind of biology. All of those have been technologies for looking at DNA, and that's just one piece of the portrait of how we look at cancers. DNA sequencing is giving us a portrait of evolution of cell populations in the tumor, but there's a lot more going on we can't see with DNA because a tumor doesn't just develop through changes in the genetic sequence of tumor cell populations. It also develops through changes in the behavior of those cells reflected in expression programs in the RNA, as well as in changes in the cell populations present in the tumor. A tumor is not just tumor cells, it is a combination of tumor and non-tumor cells that influence one another, guide one another's development, and alter one another's behavior. And we need RNA to see that. And once again, we need to really look at not just one technology or another, but bring these together through computational optimization methods that let us understand how these two different portraits interact with one another and reflect development of the tumor. Just as we are working on bringing in more complex pictures of what is happening through different technologies, we also need to work to bring in more complex models of the biology. Because the more that's learned in experimental biology about how tumors develop, the more we realize how important these different aspects of tumor development are to getting a complete picture of what's going on in their evolution. These are examples of clonal lineage trees, so evolutionary trees of tumor development that we've developed by very similar kinds of optimization methods, in this case expanding in the direction of different kinds of mutability that are important in tumor development. Early methods in this field largely looked at point mutation, so single bases that mutate in tumors, what are known as single nucleotide variants or SNVs, largely because this is computationally the easiest kind of variant to work with. Some methods, including my own lab's work, were more focused on copy number changes, so changes in the amounts of different pieces of DNA in a tumor, which require different kinds of computational theory, but are quite important in their development. And we've been working to bring other classes of variants called structural variants, large-scale rearrangements, translocations, inversions, and so on in the DNA into this portrait, because each of these kinds of variants is happening simultaneously in tumors, and each of them helps you understand how the others are working. You can't really get a complete picture of how the tumor is developing unless you look at all of these simultaneously and understand how each of these is synergistic in understanding the others. And so this has been another dimension in which we've been working. And yet other dimensions come out by bringing in information that is telling us other things about the evolution of the tumor as well as about the biology. So one of the hot areas in cancer research right now is spatial genomics, so understanding how variation within a tumor is happening within space. And we are working on this, as many are, through a variety of technologies. This is looking at some work that we're doing with a collaborator, Yangchen Lian Zhao at Carnegie Mellon's Biological Sciences Department, an expert in bioimaging, to look at how we can combine sequence and imaging data to give us a more complete portrait of how spatial structure within a tumor is affecting it. Because it's important not just to understand what tumor cells are evolving, what is the evolutionary tree, but where are they evolving? Which tumor cells are occurring where? What other tumor cells are they associated with? What non-tumor cells are they associated with? Because the spatial environment both shapes and is shaped by the tumor. As we get more and more into these increasingly complicated problems, there are whole new areas of study that I believe are becoming more important and are going to continue to become more important here. So one of the things that my lab is working on now that I think we have not yet convinced the rest of the field is important, but I'm going to tell you right now, this is going to be very important for the future of biology, is the increasing need for sophisticated computation for designing the studies that are producing the data that are going into these kinds of inferences. As we can see more and more in the computational studies, we need to look at many different kinds of modalities of data, and it makes a very big difference how you employ these. If you are looking at a tumor, we can say that you can get a much more complete picture if you have time series data from the blood and spatial data, you have single cell data and bulk data and imaging data, and that starts to create hard problems of how you pool your resources for combining these different kinds of data sources. Is it better to have twice as many patients in your study or twice as many regions of tumor sequenced per patient? Is it better to have twice as many single cells per region or twice as many regions? Is it better to have twice as many time points of blood-based sequencing or twice as many marker genes that we study at each time point? These are hard questions, and it matters a lot to your ability to extract meaningful information from the data, how you answer them. And at this point, the practice in the field is pretty much ad hoc. There's not a lot of rigor to how people ask those questions because the tools don't exist yet to ask them rigorously. And I would argue that this is going to be an important emerging area at the intersection of computation and experimental science, the increasing need for sophisticated optimization methods to pose these problems of experimental design, to come up with AI and optimization methods to solve them, as well as to solve allied tasks like how to develop simulation methods that will make it possible to solve these optimization problems, or use other methods such as automated science, which I'll say a bit about later, to make it possible to optimize over these complex solution spaces. But what all of this is getting into is that a problem like cancer biology, we increasingly appreciate, involves looking at many different aspects of the biology and many different kinds of data.
And you really cannot understand how any of these pieces work without understanding all the others and bringing in all of the kinds of data that go into understanding them. It's a process of genetic evolution of tumor cells, a process of changes in populations of these different kinds of tumor cells as well as non-tumor cells. And these two processes feed into one another. They are both influenced and influenced by, or influencing and influenced by, the spatial environment in which these processes are taking place. And all of those cells, tumor and non-tumor, are undergoing changes in their behavior that influence one another. Tumor cells communicate with one another. They communicate with non-tumor cells. And that goes in both directions, changing one another's behavior. The classic biological way of looking at a problem like this is to take one little piece of this problem and understand that as well as possible and in as much depth as you can. And I think we increasingly appreciate that just isn't going to work. You have to understand all of these different pieces of the puzzle if you want to understand how a tumor works, because they all influence one another. And as I mentioned, cancer biology is often the cutting edge of what's happening in computational biology and other sorts of data-driven biology. What's happening here in cancer is happening in every aspect of biology today. It's at different rates in some areas versus others, but I don't think anything I'm telling you about where I think cancer biology is going would be appreciably different for any other aspect of biological discovery. One other point I want to say about my scientific work and the lessons I can draw from it is illustrated by this slide here, which is that none of this happens from one group alone. When I talk about things my group is doing, it's always things we are doing with collaborators. And these are collaborators from many different areas. In some cases, it's other computational biologists, because computational biology has become a huge field to deal with all of these different aspects of the problem, and different computational biologists have expertise that we might need for different problems. It's work with experimental biologists, biotechnologists, other kinds of computer scientists. Pretty much anything happening in cancer biology depends on these interdisciplinary teams. And again, there's nothing special about cancer in that regard. That is happening in every aspect of biology, really every aspect of science today. And this is something that I think is an important lesson to take about how science is working today and how it's going to work in the future. It's a process of interdisciplinary teams, different kinds of expertise coming together. And I would argue that computational expertise is a key part of those teams, but it's not the only part. But it's a part that everyone in these teams needs to appreciate if you want to be able to work on cutting-edge science. So all of that is about my work as a scientist. And I want to divert a bit and say a little bit about my work as an administrator, because I think that reinforces some of these same lessons I've been trying to get across as I talk about my own lab's work. To a large degree, becoming an administrator means thinking about the field more broadly, so trying to represent a whole department, or when I work with my professional society, the International Society for Computational Biology, representing the whole profession. And that really means bringing in, seeing how these same kinds of lessons play out well beyond my own research agenda. So I do have the privilege of being head of Carnegie Mellon's Ray and Stephanie Lane Computational Biology Department. I don't know if everyone, all of our Carnegie Mellon community is aware that Carnegie Mellon has one of the best, if not, I would say the best computational biology departments in the world. And I think it's been a great privilege to be head of that department the past few years. And I think the reason it's been such a successful department is because, first of all, we take a very broad view of computational biology. We have recruited a group of scientists who cover many different areas of computational biology, as well as many different kinds of computing that go into them. So this is our faculty. So 2 members have not officially started, but will within about a month and a half from now. But we've recruited a computational biology department that covers many kinds of biology and that covers many computing disciplines that go into them. Not only that, we've recruited a set of faculty who see themselves as leaders of different areas. We're not looking for people who want to be in computational biology to be service people who are handed data and figure out and are tasked with solving a particular problem on that data. We recruit people who are creating new fields of science, figuring out how to use computational thinking to ask new questions, do new kinds of discovery, and work with their own teams of collaborators to make new science possible. We are not unique in having a computational biology department, although this is an unusually large group for one of these, and we are quite unusual in having a computational biology department in a school of computer science, which I think is another reason our department has had unusual impact. We believe in the importance of computational thinking and in innovation in computation as a way of driving biomedical discovery. So we've been able to create a department that has a number of different strengths in it, some of these being methodological strengths. Our department was founded with a belief that artificial intelligence and machine learning would be key technologies for the future of biomedical discovery. That was a very prescient decision from my predecessor, Bob Murphy, who founded the department. And it has proven to be a great decision because pretty much anyone working in computational biology today is doing AI and machine learning, and anyone working in biology today is probably aware of AI and machine learning having impact within your field. So that has been something that has been quite successful in our department, but we also have experts in discrete algorithms, statistical computing, and so forth. Another area of focus of the department from the beginning has been automated science, and I think this is another place where there is going to be a change in the field, increasing dependence on automation and robotics as a way of doing scientific discovery. Automated science is really about AI-driven robotic laboratories that are running their own experiments, doing their own discovery, not just running experiments planned by humans, but using AI to gather the data from those experiments, synthesize it, and plan their own future experiments. And this is something I'll say a bit more about. But we've also been able to develop areas of focus in basic biology. So much of our department is working one way or another on single cell or spatial biology from many different directions, as well as more applied areas such as biomedical applications in cancer research like my own lab, but also diseases of aging, neurodegenerative diseases, infectious diseases, genetic diseases, and so forth, all of which are areas that are depending today on computational discoveries. So to give just a few quick examples, within our department, we have people working on genetic variation and evolutionary biology from the single cell scale that I work on to scales of variation within populations, like within the human species, variation between species, and even variation within multi-organismic populations. We have people working on aspects of computational disciplines, in this case, image analysis, from scales from the nanometer scale to the subcellular, to the cell type, to the tissue level, bringing in many kinds of insights and bringing these to many different kinds of biological discovery. We have faculty working on genetic regulation of different kinds, from transcriptional to chromatin structure to protein-protein interaction, and again, it scales from the single cell to the organismal to the population to comparative genomic methods across species. And we have special focuses such as automated science, as I mentioned, with faculty bringing these kinds of technologies to different problem areas, for example, genetic regulations, single cell imaging, as well as pioneering allied areas such as AI for designing algorithmic parameters and problems such as study design that I told you a bit about before. Our faculty are not just having impact on computational biology, though, which is one of the important lessons I want to get across here. They're having impact on experimental biology quite broadly. So among our faculty, we have leaders of a number of different U.S. national or international research consortia that are consortia combining experimental and computational study. So these are consortia working on problems such as aging, genetic regulation, single-cell genomics, comparative genomics, and organismal evolution, where Carnegie Mellon computational biology faculty are playing important roles in these large consortia, leading initiatives for things such as computational tool development and data analysis. And my point here is just to say that computational biology has become important across pretty much any kind of biology you can think of, and building strength in computational biology leads to impact across these many fields. And that is a trend that, again, is true in our area and is going to be true in many other areas. This includes Carnegie Mellon's pioneering work in automated science, so developing automated robotic laboratories embodied by what was called the CMU Cloud Lab. So this is a large warehouse-sized facility back home in Pittsburgh, back from my home, that is doing this kind of automated science on a massive scale, and I think it is a representative more and more of how science is going to be working in the future. We also have a large educational agenda, research educational programs at every level, from doctoral to master's to undergrad to even high school level, specialized programs such as increasing numbers of MD PhDs training, doing their PhD in computational biology or allied disciplines, specialized programs such as automated science. And these students are experiencing tremendous success because there is an incredible demand for students in these areas, an incredible demand for graduates. And this includes large numbers of PhDs. I think at this point, we have about as many PhDs graduating in computational biology as we do in biological sciences in Pittsburgh. And I think that reflects the increasing need and demand for students with this expertise, and growing numbers of students doing minors, additional majors, concentrations, and so forth. For example, doing experimental biology, but knowing that they also want strength in computational biology because that's the direction the field is going. And what all of that is telling us is that as an administrator, I can see that what I work on in my own niche area of computational biology is not in any way special. Again, this is happening in every aspect of biology across these many different disciplines we work on. And that leads to one last area I wanted to briefly mention, and that is my work as an educator. Because I have been working for many years as an educator, trying to teach both computational and experimental biologists what they need to know about computation, working on planning at the level of degree programs. How do you design a degree in computational biology at any of these different levels? And increasingly with a global community of bioinformatics educators who are working on trying to define standards for the profession. So trying to identify what it is that anyone needs to know about computation to be working in the life sciences. And I will just put in a plug. I'm not just here to visit CMU-Q. I'm also in Doha because there's an international conference on bioinformatics education happening this Wednesday and Thursday in Doha at University of Doha for Science and Technology. So I hope any of you interested in this field might have a chance to check that out as well. But just to very briefly give you some perspective on why it is that we're working on this, I want to say a little bit about some work that I had nothing to do with. And that is represented in two different documents I'm highlighting here. The first of these is something called Bio 2010. And anyone who's been around biology education for a long time has probably heard of this because it was a very influential report that actually came out, I believe, in 2003. The name Bio 2010 was aspirational, a projection of where the people developing this report felt biology should be in the future, back when 2010 was the future. And one of the key conclusions they drew from this was that biologists of the future were going to need to know about quantitative methods. Biology was going to be a much more quantitative and more computational discipline in the future than it was at the time in 2003. And they put forth this conclusion that the way biologists were trained was going to need to change to catch up to that. And so this Bio 2010 report put forth the aspirational hope that by the time we got to 2010, every biologist would be learning about how to do computing. They would have enough competency in computer science to be doing sophisticated data analysis, modeling, and so forth. I highlight this other report because it was a study that came out in, I believe, 2017 that was basically asking the same question retrospectively. Where was biology at this point? How much progress was made in getting to the kinds of things Bio 2010 said were needed? And I'll just give the punchline here. Their conclusion was, I'm putting words in their mouth, again, this was not my study, but their conclusion was there was basically no progress. Pretty much nothing had changed between 2003 when this report came out and 2017 when this one came out. Biologists were trained in pretty much the same way they had been 14 years earlier. And they came up with a lot of reasons why this was the case, but from my point of view, basically the conclusion was nothing much had changed. And that is part of what inspired myself and many others in the field to try to ask, how do we move the needle here? How do we actually lead to change in this field? How do we figure out how to reform education across the life sciences, not just computational biology, but experimental biology and allied areas like the health sciences? That led to a large community of research, a number of different reports. The latest is now in press on setting standards for training in this field, as well as a growing community of educators working in this area. This started from a very basic question of just asking, what should we be teaching biologists or computational biologists? What do you actually need to know about computational biology to be working in the life sciences? And this started from a little committee, just 3 of us at first, Lonnie Welch, Fran Lewitter, and myself, trying to figure out what were the standards of the field. We set out to look at this by looking at what employers were looking for in hiring, as well as what degree programs available at the time were offering, trying to figure out what were the standards of the field, what was the practice that we could recommend everyone else adopt. We came up with different ways of quantifying what were different programs offering and tried to ask, what do they have in common? So we could come up with some recommendations for the field as a whole. And once again, I will just jump to the punchline here. What they had in common was nothing. There was nothing that you could find that a majority of programs trying to teach computational biology could agree should be taught to computational biologists at that time. And that really led to a tremendous amount of work to try to figure out, if there's no agreement, what should there be agreement on? Work of trying to set what are called core competencies, so trying to go to the community and say, okay, what can we agree on? Moving to a higher level of abstraction to try to get at some notion of what it is a computational biologist needs to know, as well as a notion of the different communities that need to know about computational biology, what were called personas, describing different people with needs in the field, including experimental biologists who might be using this primarily while doing primarily experimental work, to people that we call bioinformatics engineers, people developing new tools in computational biology, and then going out to the community and trying to engage many educators in the field with trying to advance this, to figure out what should we be teaching, what levels do we need to know these, how do they need to be taught within different populations, coming up with finer and finer categories for what a bioinformatics educator should know, and increasingly refining these to, in the latest 3.0 version, through growing bioinformatics education community, to a more advanced notion of what these competencies are, as well as much finer detail of what's going into these, so we can start to at least make some sort of prescriptive advice of how you should be learning bioinformatics. We've been able to put this into different kind of processes, so a working process now for being able to evaluate different degree programs, as well as a continuing education community that is using these kinds of competencies to study how we can assess programs, how we can improve them, how we can continue to develop the competencies and so forth, as well as bring this into the future of looking at some of the grand challenges of the field and how these are evolving over time. So to try to come to some conclusions, what did I learn from all of this? Well, as a scientist, I learned that modern science requires an interdisciplinary approach, but computational thinking is a key part of this. And being an administrator taught me that this is not just something true of my own corner of the field, but something that needs leadership from computational thinkers in every area of biology, and that this is creating hard problems of how you provide training to get there. So what comes now? In the past, biology was largely a study that might be represented by this illustration from DALL-E 3 of what a biologist might have looked like if we imagined them 50 years ago. One person looking in a microscope, trying to figure out how biology works. It was largely qualitative. We didn't really have a concept of what today we would call rigor and reproducibility. And today that things are better. Experimental and clinical scientists need to know about statistical rigor. They have some training in these areas. They know how to use at least off-the-shelf tools. But at the same time, the field has really outpaced the training there. It's really outpaced what is the standard in biology education, to the point where also at the present, we have these vastly complicated multimodal problems that require much more sophisticated computational thinking than is the standard of training today. In the near future, I would argue that this has to change, that anyone working in the life sciences is going to need a much more sophisticated knowledge of computation and computational tools to be able to work successfully in this field. And I think that is only going to become more dramatic as we go further into the future. I believe a lot of the future is going to increasingly depend on automated systems, increasingly depend on AI, and depend on new directions such as AI-generated protocols, open libraries of experimental protocols, and increasingly computationally driven design of experimental systems and data interpretation that is going to require a new kind of scientist that really doesn't exist yet today. And it really is going to come down to all of us to figure out how we do this. How do we build the infrastructure, the scientific theory and tools to make this happen? But more than that, how do we partner to make this a part of the practice of science? This is not just a research question, it's a cultural question, and it's an educational question of how we get the field to where it needs to be. And I think it's become a more and more urgent question throughout my career. So I know we're running late here, so I just want to wrap up by leaving you with some of these challenges that I am working on. I hope many others of you will be working on about how we get the field to where it needs to be in the future. I'll just say before I close that what I told you about on the research side is work of many people in my own lab, labs of collaborators, funders, and sources of data, and the educational work is no different. It's the work of a large community of educators who has come together to work on these kinds of problems and help us come up with better solutions to them. So thanks to all of them, and thank you to all of you for your attention.
All right. Thank you very much, Russell. That was a very interesting talk. Does anyone have any questions? Yes, please.
First of all, it's very interesting talk. Thank you so much.
Can you turn on your microphone?
Yes. Okay. So my first question is about quantum computing. There's been a lot of talk about quantum computing and the future of quantum computing. Is this something that there are efforts to, like, as you said, like the biology or the science follows the technology. So are there efforts to kind of integrate quantum computing in computational biology?
Yes, there are. It's not my area, so I'm not the most expert person to tell you about it, but some of the work I'm aware of, a lot of it's been successful in the more biophysical problems where quantum computers can basically work as analog computers for solving quantum calculations important to structural biology. But that's not how most people would say we should be using quantum computing. That's one place where they are particularly powerful. But also other areas such as quantum computing for deep learning. So that is a very active area today, trying to use quantum computers to accelerate the kind of deep learning-based AI that's becoming influential through many areas of computer science, including biological discovery. So this is an area I know many people are betting on becoming much more important as we go through the future, but these are some of the things I'm aware people are working on right now.
Is this something dynamic? Is this something that's also taught in CMU, like for computational biology?
Well, I'd say within Carnegie Mellon, we don't have anything in computational biology that's teaching quantum computing. There is quantum computing in other areas of Carnegie Mellon, but not that I know of collaboration with computational biology. So it's a place where if one of my students needed to learn about this, I would be sending them to others working on quantum computing, whether that's in our physics department, in our theory of computing group, or so forth. So you can learn it, but it's not taught by computational biologists at CMU. No, I believe our deadline is, I'm not sure the exact date, actually. I should probably know that, but it would be around the beginning of December, so soon, but not too late to apply.
There was another question there.
Hello, thank you for this comprehensive presentation of the field. I have a question. Where do foundational models and generative AI fit in this research program?
Yeah, so this is another area that I think the field is very excited about. People are using foundation models for a number of areas already. So, for example, the automated science I told you about, there's been some very interesting work actually by a member of the chemistry department in Carnegie Mellon on using foundation models to help generate experimental protocols, automatically design experimental protocols for automated science. But there's a lot of excitement about essentially foundation models for biology. So, for example, the foundation model of cell biology. I have colleagues working on trying to generate that kind of model for understanding
all the different aspects that go into how a biological system functions and all the different kinds of data that bear on them. So I think there are uses of the foundation models that already exist in biology, but also a lot of interest in developing new kinds of foundation models specific for biology.
Maybe one last question. Ihab.
Thank you, Russell. I'm right here. I'd like to just focus again on the educational part of your talk. I couldn't agree with you more that we need to change the way we educate biology students and add the more computational part. It has to go beyond teaching like one course in computational biology. I think it has to be an integral part of the curriculum. But how do you train the current educators who none of us is actually trained like this to train the next generation to become more computational thinkers?
Yes, and that is a challenge. And if you remember the paper I referred to about barriers to bringing computation into life sciences education, that was one of the barriers that they identified, that most life sciences educators don't have a lot of background in computation. So I think some of that has to come from new people coming up in the field being trained differently. It just takes time for this to propagate, but you start with the trainees today and train them to a different standard than perhaps their own professors, and then they will become the educators of the future. But for people in the field, there are a lot of ways to learn, a lot of resources, including colleagues, online resources. So there are actually one of my colleagues in computational biology, Philip Campo, has a tremendous set of online freely available resources for anyone who wants to learn about the field. And all of us as scientists are constantly learning new things. I don't think computation is fundamentally different from having to keep up with our field in many other ways. So I don't have easy answers, but I think it's something anyone working in the field should be learning more about if they don't have a background yet.
Thank you very much, Russell. For others that have questions, there's going to be a small reception outside. I would like to invite Dean Michael Trick to close the session.
Thank you. How about another round of applause for our speaker today? Fantastic talk. It looks like we've got some possibilities to add to your lab, and so obviously very inspirational. So thank you for coming out and joining us today. As a small token of our appreciation, we do have an award for you, and so if you want to come over to me, they're going to be putting up something there. I'm going to have to put this down just a second. And you hold this and you get the right background. Wait one more. Good. Thank you very much.
Thank you.